transcriptional control is controlling whether the gene is transcribed at all. And we talked about that last time. If you put methyl groups on it, on the DNA, it tightens up and you don't have any transcription. Can't happen when it's all tight and coiled up. Remember we talked about that? So that's a methylation and acetylation. Those are examples of transcriptional controls, controlling whether transcription happens. All the transcription factors that will attach to the promoter at the beginning, those are transcriptional controls. So we can control whether we even start transcription or not. Now, in a eukaryotic cell, there are a whole bunch of transcriptional controls. Look at these. This is the DNA here, okay? So the DNA, this orange strand that you're looking at here is DNA. And here's the gene, and here's the promoter. RNA polymerase attaches to the promoter. But it cannot do so unless there's a whole bunch of different transcription factors in place. And this is showing all these circles here, all transcription factors that all have to be attached to the promoter and just right. And there's even a DNA bending protein, which is this purple thing right here, that makes the long DNA strand bend around on itself. And there's a site far away from the gene, it could be thousands of nucleotides away, that have these, it's called an enhancer site. And it has other proteins on it called activators, which will, the whole DNA loop will kind of bend over here and turn over. My remote control mouse isn't working so well. That makes me very unhappy. Anyway.
Does it need new batteries?
I don't know if it should be charged. It might not be charged enough. Feels like the computer is being slow. Okay, so anyway, the DNA can bend all the way over like this, and there are proteins that attach to this enhancer site that have to bind with proteins that are attached to the promoter site before this RNA polymerase can start transcription translation. So there's a lot of things that have to be in the right place to get this going. And so that's what these videos are about. So check these out. Absorb the information.
By controlling which genes are active, a cell can take on special characteristics. Muscle cells and neurons have the same DNA but perform different functions because they use different sets of genes. A key challenge in genetics research is to understand how transcription factors find the correct DNA sequences to control genes. In the nucleus, DNA is condensed into a DNA-protein complex called chromatin. In places where genes are being expressed, there are often zones of naked DNA. We will now explore ways that transcription factors interact with these zones. Even among scientists, a common misperception is that transcription factors fall from the sky onto the DNA and test one spot at a time. Such a process is too inefficient. Instead, the transcription factor interacts non-specifically with the DNA backbone. This allows it to efficiently search the DNA for its binding site, a DNA sequence it recognizes. The transcription factor slides along until it encounters its binding site. Then it changes conformation to interact specifically with nucleotides on the interior of the helix. Now the transcription factor can stay and interact with other proteins for transcription. Or it can become dislodged by many factors, which can include movement of the DNA or interaction with other proteins. Studies have suggested that transcription factors sometimes move from one part of the DNA to another. Short hops might allow them to bypass other proteins or to hop over compacted sections of DNA.
Y'all put the Skittles up, please.
When the transcription factor lands on another segment of DNA, short hops become big jumps. Although the other strand is nearby in three-dimensional space, it may be much farther away on the chromosome or on another chromosome entirely. There are 1,500 different types of transcription factors in human cells. They interact to create a complex language of gene expression.
So all those little things they were showing jumping around on the DNA, those are these things. They'll slide along the DNA until they find the promoter, and then they'll stop and attach there.
That's rad.
One more video. Kind of shows this thing happening.
Provides an overview of the major steps involved in RNA production. mRNA and eventually proteins are produced when and where they are needed in an organism. Some genes, such as those for housekeeping proteins in processes like glycolysis, are expressed all of the time. These genes undergo what is known as constitutive transcription. Other proteins are only produced at specific times in certain cells. In these cells, genes undergo a process called regulated transcription. Transcription is regulated by proteins called transcription factors. These factors are proteins that are produced in the cytoplasm and eventually migrate into the nucleus, where they interact with DNA and activate transcription. These transcription factors only interact with specific genes, those genes whose transcription they control. Most eukaryotic species have over 1,000 transcription factors. Regulated transcription begins when a signal is received by the cell. This signal, often a protein, begins a signal transduction cascade that lets the cell know certain proteins are now needed. The most common activation event for a protein in a signal pathway is the addition of a phosphate group. That activated protein in turn interacts with another protein. This interaction leads to the phosphorylation of the next protein in the pathway. There is often a series of proteins that are activated by phosphorylation. At the end of this cascade, the final protein will be activated and then enter the nucleus. It must make this journey because transcription only occurs inside the nucleus. After entering through a nuclear pore, the protein interacts with the specific transcription factor responsible for activating mRNA production. As with the earlier proteins, the transcription factor is modified through phosphorylation. The activated transcription factor will next bind to an enhancer region. The enhancer is a region of DNA upstream of the transcription start site that binds a transcription factor. The transcription factor then binds to the DNA and moves to interact with the rest of the transcription protein complex, located at the transcription start site. When the formation of this complex is complete, transcription of the gene will begin. It is this sequence of events that ensures genes required for a specific tissue or only at a specific time are expressed appropriately.
Man,
Why does the DNA need to pinch like that?
it's a good question. It probably doesn't need to in every case, but it does often. And it's just another control. You know, it's another way of turning it on or turning it off, another thing that has to be in place. So it's just very important not to be transcribing genes when you don't need the protein. What a waste of energy, because every time you make a protein, it's using a lot of ATP and that kind of stuff to put it together. and you don't want to waste that. So if you have some sites on it, that'll stop it. These transcription factors will stop it, and apparently this site upstream on this one stops it too. If you have a bunch of sites that'll stop it, that'll keep it from being made when it shouldn't be. And I guess that's how they evolved.
Got it.
But it's a good question. I bet for a lot of them, we're not sure exactly why they have so many. It's good to keep them from not doing it. So all of that we just learned, all that section, those are transcriptional controls. Controlling whether transcription starts at all. Let's talk about some post-transcriptional controls. After transcription, what do you have? What have you made when you transcribe a gene?
mRNA.
You've made mRNA. Are y'all listening? Only a few people even transcribed on that one. After you transcribe a gene, you've made mRNA. Y'all remember? We already learned this. What has to be done to the mRNA before it can leave?
Sure.
You got to cut out the introns. What else do you got to do?
Guanine cap.
You got to put a modified guanine cap. You got to add some adenines on the back. Polyadenine tail. Adenine. 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 Dropped an adenine. And all of this would be considered, because it can't leave the nucleus till all that gets done. All of that's considered post-transcriptional control, because it happens after transcription, right? This shows you can take the pre-mRNA that has the introns in it, and you can remove the introns. In this case, they removed all 3 introns, and there's genes 1, 2, and 3. Now this one over here doesn't include number 4. So we call this alternative splicing. The spliceosome protein can just remove all the introns, but it could also remove all the introns and remove one of the exons. It could cut out that intron, and that intron, and what if it also cut out that exon?
That would be bad.
Well, not necessarily. It could make a different protein. It would make a protein that's shorter, for instance. So this is a way the cell has of making different proteins. If it left number 4 in, it would make a longer protein. If it cut number 4 out, it would make a shorter protein, one that's different.
Does it do it on purpose?
Yeah, it can do it on purpose sometimes. In this one over here, look, it cut out exon number 2. Exon number 2 is not there. So that could make a different protein. And very often this is done on purpose, because what scientists figured out was that there are about 40,000 different proteins in a cell. But there's only about 10,000 different genes. So how can 40,000 proteins be made from 10,000 genes? This is how. Alternative splicing of DNA. Many of the genes can code for multiple proteins by splicing out some of the exons when you do the splicing. You can get totally different proteins that do different things. You look confused.
Well, I'm saying that they're like, are they doing it, will it happen by mistake sometimes and then they make a wrong protein?
I guess it could happen by mistake, but often it happens by design, where you cut out certain introns, you cut out one intron, you cut out one exon and you get a protein that has a different shape. It's maybe a little bit shorter. If you leave that exon in, you get a different protein. So by changing which exons you cut out in addition to the introns, you can get all sorts of different proteins. And they show all sorts of different things you can do. In this picture here, they've cut out the gray are introns. They've cut out that intron, they've cut out that intron, and you get the bottom one. But if you cut that exon out, you get this mRNA. So there's all sorts of different ways you can take a piece of mRNA and cut it. You see, in this case, this is called, and you don't have to remember all these types. They'll show it to you in any questions that they have. But look at this. Mutually exclusive exons. Here's the piece of pre-mRNA. In this one, in this top one, they've cut out all the introns, and they've cut out the red exon. And you get this mature RNA. In the bottom one, they've cut out the yellow one, and you get a different mature RNA. So you can make different proteins. These are just many different examples of ways they can cut out exons differently. And you don't even always have to cut out the introns. Why not leave the introns? We call that intron retention. So you could cut out this, if this was the intron, you could cut it out and remove it and make that mature mRNA. Or you could just leave the intron and get a different mature mRNA. In that case, it's not even an intron, it's now an exon. Your introns could turn into exons. So there's all sorts of post-transcriptional modifications. Things you can do, if something happens to the mRNA, you can modify it, change it around in a way, cut out introns in a certain way, and you can change the way the protein turns out. You're not changing the DNA. The DNA stays the same. You're changing the mRNA. Here's another post-transcriptional control. There are segments called UTRs. At either end, there's a segment called a 5' UTR and a segment called a 3' UTR. This is on the mRNA. Here's the guanine cap, and here's the 5' UTR. UTR stands for untranslated region. So that region is not going to be translated into a protein. So what's it there for? It's a piece of mRNA that's not translated into protein, that's there for things to bind to. Proteins can bind to it, and also on the 3' untranslated end, proteins can bind to it and maybe shut it off. Maybe make it where it's not going to be translated at all. Or, they're called, by the way, they're called RNA binding proteins. Here you see 3 RNA binding proteins bound to the 3' untranslated region. Maybe they bind there and stop the thing from getting translated. Maybe they bind there and make the thing get translated faster somehow. There's all sorts of different ways the mRNA can be affected. And one way is with these UTR, these untranslated regions. They're binding sites for more proteins. This is exciting stuff.
Yeah.
Mm-hmm.
Can you believe there's so many ways to control the...
Are there any mutations that go with this?
Whenever we say mutation, we're usually talking about the DNA. So you don't really talk about mutation once you make the RNA. DNA, the mutation is a change in the DNA. So, and that's a common, students will confuse that a lot. They'll say there's a mutation in the RNA. The mutation happens here.
Yes.
These are pretty cool, MicroRNAs. They're short strands of RNA that are produced by a gene, and they'll actually coil up on themselves like this. Coil back on themselves and make kind of a loop. You see how that's kind of coiled back on itself? It's called a microRNA. And what it does is it destroys other pieces of RNA that are in the that are floating around in the cytoplasm. MicroRNAs are what we call RNA killers. Would you like to see how one destroys
Very much.
RNA, I have video footage. Let's kill some RNA, shall we?
Yeah.
Dark was Smarty, 16.5 minutes.
Yeah, we're running through the end of 16 today.
Almost.
Yeah.
This is a cell, the basic unit of all living organisms. Our body consists of different cell types, like skin, muscle, or bone cells. They have different structures and functions, but contain similar components, like the nucleus. The nucleus contains the DNA that encodes the instructions for cell development and function. This information is arranged into segments of DNA called genes. Almost every cell contains identical copies of DNA. But if there are the same instructions for all cells, why are there so many different types? The reason is gene silencing. Genetic information can be switched off. So, during development, a cell only reads instructions that are necessary for gaining the characteristic structures and functions. For gene silencing, a cell can use a microRNA. This powerful molecule is able to turn off genes by inactivating messenger RNAs, which are necessary for translating the genetic information into proteins. In this way, genes can be turned off. MicroRNAs participate in the regulation of the cell from its development to its death. Their dysregulation can have serious consequences for our body and can cause a range of diseases such as cancer and heart disease. Having such a big impact on our life, microRNAs deserve a closer look. So, let's start at the beginning. Similar to proteins, the genes coding for microRNAs are contained in the nucleus in the DNA. Each gene is transcribed by RNA polymerase II, which produces either a regulatory or a messenger RNA. In this case, the transcript is a primary microRNA, which forms a typical hairpin loop structure. It will become the final microRNA with regulatory function after several steps of processing. First, the double-stranded stem is recognized by the protein DGCR8. An enzyme called DROSHA associates with DGCR8 to form a microprocessor complex, which is able to cut the RNA into a smaller precursor microRNA. It can now be exported into the cytoplasm, where it will inactivate the messenger RNA of one or multiple genes. The precursor microRNA is carried out of the nucleus through a nuclear pore by the transporter molecule X14-5. In the cytoplasm, it is recognized by a large RNase protein called dicer. Dicer cleaves the stem loop and forms a short double-stranded microRNA molecule. In the next step, an Argonaut protein AGO2 interacts with dicer to bind to the microRNA. The microRNA is unwound and one strand is released. The remaining strand, called the guide strand, interacts with AGO2 and some additional proteins to form the RISC, the RNA-induced silencing complex. It can now be guided to its target and inactivate one or multiple genes. The messenger RNA of a target gene is complementary to the sequence of the microRNA. That enables base pairing. Once bound, there are two ways in which RISC can inactivate the mRNA. Proteins in the complex can simply cut the messenger RNA, which will be further...
So if you're not following here, if you got lost, this is a piece of RNA that's in the cytoplasm. The microRNA is going to come attached to it and disable it, basically. This is telling the ribosome to make proteins, but it's going to be disabled.
It is complementary to the sequence of the microRNA.
There's the
that enables
microRNA.
base pairing.
Bam.
Once bound, there are two ways in which RISC can inactivate the mRNA. Proteins in the complex can simply cut the messenger RNA, which will be further destroyed by the cell. Inhibition of translation is another mechanism. In this case, the RISC complex prevents the ribosome subunit from binding. In both cases, the messenger RNA will not be translated into a protein, and the gene is silenced. Since their discovery in the 90s, major parts of the microRNA's pathway still remain unclear. However, with their essential role in many biological processes, microRNAs offer great potential for medicine and might lead to key treatments of various diseases in the future.
Yay!
Mr. Willis.
Yes.
Are we skipping 16.7? That's what it
It
says
says
on
that
my
way.
web.
Yep.
Yeah.
Is that okay?
Yeah. I
No,
just-
no, so.
Yep. Skip 16.7. All right. Let's talk about translational control. How are you going to control translation? Where does translation happen in a cell?
The cytoplasm.
Where in the cytoplasm?
Ribosome.
The ribosome. So if anything happens with the ribosome, that's going to be a translational control. And here it shows one example. There's this protein called EIF2. And I always thought it was ELF until I started teaching this, but it's EIF. And that stands for something, something factor, initiation factor. Anyway, what happens is this EIF2 protein, and what I need is a magnet. This is an EIF2 magnet. But anyway, it'll come, pretend this is it. It comes and it attaches to the ribosomal subunit. It attaches to the small subunit. Boop. And that keeps the ribosome from being able to translate. It'll have a phosphate group attached to it. I need a little phosphate group too. But this EIF protein, when it's phosphorylated, it's the right shape to attach to this thing and shut down translation. And so no RNA will be able to go through this when this EIF protein is attached. If the phosphate comes off, the EIF protein can unattach, and you're able to do translation. So that's a translational control. And it's all this protein, EIF, that attaches to the ribosome. There's a bunch of others. There's other proteins that can attach to ribosomes and shut them off. This is just one of them. And take a look at it. We've got out of time, aren't we?
The basic steps of translation, including initiation, elongation, termination, and recycling, are the core steps that happen in order for an open reading frame in an mRNA transcript to be translated into its encoded polypeptide. RNAs are a key signal of nutrient deprivation as tRNAs become uncharged when there are known as REL-A, which when bound in this state promotes the synthesis. Eukaryotic cells also respond to uncharged tRNAs in the cell, but the response is different at the molecular level. In eukaryotes, the uncharged tRNAs do not bind to the ribosome, but instead bind to a kinase known as GCN2, which has a
No, that's not the one I want. Oh, here's EIF. There, see, EIF2. There it is. And it's phosphorylated, it looks like. And it attaches to the ribosome. I don't know, we don't have time to look at that, sorry. I didn't quite finish, I didn't get to the post-translational control, but I will show you that in one slide tomorrow. And you could have a quiz, so make sure you finish this chapter. You don't do 16.7. And who wiped last week? Oh, I already said who the wiper